Hello, this is Nick Cap, and this is General Microbiology, and we will be talking to you about the historical background of microbiology. And let's see if we have my cursor on here, and we're ready to go, everyone. Okay. First thing off, we got to look at what we call binomial nomenclature, and this was invented by Carl Linnaeus in 1735. Each organism has one specific name contained in genus and its species, which is specific epithet or genus name. The first letter is always capitalized when you write it down. The species name is always lowercase. Escherichia coli is an example of one of those that you need to do. And here's an example of a microorganism that we'll hear a lot about in this class. And so again, naming things scientifically is understanding them. Escherichia coli was named in honor of its discoverer, Theodore Escherichia. Described the bacterium's habitat, the large intestine, or the colon. Again, coli. Staphylococcus aureus describes the clustered staphylospherical cocci cells, and it's kind of golden in color. Scientific names after their first use, when you're looking at them, they can be abbreviated. So Escherichia coli and Staphylococcus aureus can be found in and on the human body. And there, we have E. coli is found in the large intestine. S. aureus is on the skin, although they can both be in both places. And if we notice, we can kind of simplify this by looking at that. You will be looking at these in a scientific paper. And so again, a test that we'll look at is things like Baker's yeast, which is the correct way to write this out. And you're going to write this down and give it to me. But basically, it should be Saccharomyces cerevisiae, important here. And so that's it's understanding for us. It's important for us to understand kind of how we do this. Important events on here. And again, this is something that we're going to be looking at, and you're going to be developing for the class. Robert Hooke, 1865, described the cells. Antoine van Leeuwenhoek developed a way to look at microorganisms underneath a microscope on it, a number of things. And so here's an example of Leeuwenhoek's thing that he did. There was a spontaneous debate on what we call spontaneous generation. Spontaneous generations where living organisms arise from non-living matter and vital force of life forms. Biogenesis, living organisms arrive from pre-existing life. And there was a debate on this for a long period of time, and this is 1660s. Reddy filled 2 jars with decaying meat, and 1 jar was unsealed, 1 jar was sealed. In the unsealed jar, maggots appeared, and in the other jar, there was no maggots. It doesn't mean that the meat was preserved. It still rotted, but their life wasn't there. And so, again, where did the maggots come from? What was the purpose of sealing the jar? And we'll have a talk about this. Again, John Needham, Lazzaro Spallanzani used nutrient broth that was heated, then they placed it in a sealed flask, and we saw no microbial growth. Again, where did the microbials come from? What's spontaneous or neogenesis? Nutrient broth placed in a flask, heated, and then sealed. No microbial growth. Again, we're kind of looking at these important events that we're looking at. Rudolf Virchow, 1858, arise from all cells alive from pre-existing cells. Louis Pasteur in 1861 did this nutrient broth placed in a flask, was heated, not sealed, and he was able to just kind of actually see no microbial growth in there. It's just spontaneous generation or biogenesis. And he used what we call a swan flask. And if he took his broth in here, heated it up, and left it in a curved here, and then when he opened it up, we got microbial growth inside of there. 1857 to 1914 was the golden age of microbiology. And I believe that we're having a golden age of microbiology kind of right now, and I'll talk about that a little bit later. This is beginning with Pasteur's work. Discoveries included the relationship between microbes, disease, immunity, and antimicrobial drugs. Fermentation and pasteurization. Fermentation is the conversion of sugar to alcohol to make beer and wine by microbes. Microbial growth is also responsible for spoilage of food. Pasteur demonstrated this spoilage bacteria could be killed by heat and was not enough to evaporate the E. coli in the wine. Pasteurization and the application of high heat for a short time. It's a process that we typically use now. The germ theory of disease. was kind of looked at by Joseph Lister and Robert Koch. Basically, you take a healthy organism, you can look at its blood, and when you try and plate out microorganisms, they're not present. Again, in a diseased organism that's dead, in this case here, you can see the microorganisms that are in there. If you culture these out, you can see the microorganisms, and if you inject them into an animal, they're dead. And so this is what we call Koch's postulates, and we see that they have to be working. This process that we call vaccination is, again, Edward Jenner was working on a number of people, but he inoculated a person with cowpox virus who was then protected from the smallpox. This is vaccination from vaca, meaning cow, and this production is called immunity. Fortunate accidents, we're looking at antibiotics, and so here's a mold here that produces antibiotics that do not grow. Modern developments in microbiology, we have a number of these ologies, these studies of, and this is bacteriology, mycology, virology, parasitology, and immunology. Again, here's an example of how beautiful microorganisms can be, and actually this is the egg of an insect on a little piece there, and there's microorganisms. Next thing we're going to look at classification of microorganisms. Again, how do we name these? And we have a better understanding of these if we understand kind of what they are. What are the three domains? List some of the characteristics of the three domains, and we're going to be going over that in class, and we'll have an activity on this. All right, so thank you very much.